Okay, so it took me a little bit to get this organized. Originally this was done on February the 2nd. 2012 at 12:29 p.m. Today is February the 4th at 12:33 a.m. This is part 105, and it's line 18A3 and then A. Line 18A3 has a lot of videos that are going to go with it. It's already up to A3H, just so you know. So I do the letters of the alphabet after the A3 for each video, okay? So this is line 18A3A, Reactor, Vessel, Cell Cult, NASA Arms, ADSEP, Shuttle, STS-95, GENIA, GENIA, Wow Seti. Okay. The second part of this video is on line 18A3B, Artificial Gravity, Centrifugal Force, Centrifugal Coriolis Forces, Linear Acceleration Velocity 1g, UFO Speeds, Wow Seti. This is based on the Wow Seti 1977 radio alien signal. That's the equation found on that line, and when I googled 823.5294, certain parts of data came up in 1918A, and I've pulled all those parts and expanded them in these videos here. So the Google searches continued from data found in line 18A. Cell culturing cell cult is an automated cell culture laboratory housed in an advanced space experiment processor, ADSEP. Brief facility summary. Cell culturing cell cult is an automated cell culture laboratory housed in an advanced space experiment processor, ADSEP. It contains one rotating Reactor vessel that is fed fresh medium from a nutrient bag in perfusion, batch, or sampling mode. Up to 6 samples of medium or cells can be collected during a flight, and if desired, the entire culture can be fixed before it's returned to Earth. And I got that from the www.nasa.government mission site. And the point of contact is Paul Todd, or ptodd at techshot.com. Next is previous ISS missions. Cell Cult has flown on Space Shuttle flight STS-95. So I googled it to see what it was. Now this is photos of NASA Space Shuttle Discovery STS-095 and 096 astronauts and what a cell cult is from the Advanced Space Experiment Processor. And I just screwed that up. Sorry. Let's go back here. Try not to use the mouse too much because it's hurting my arm. So this thing here is the Space Shuttle Discovery STS-095. That's what it looks like inside the cockpit. And that's at www.aviationspectator.com. And there it is going on the road there. STS-96 Space Shuttle Discovery rolls back into the vehicle assembly building photo. Credit NASA. Okay, these are the astronauts that were on the STS-96 crew photo with Commander Kent Romner, Pilot Rick D. Husband, Mission Specialist Alan Okna, Tamara E. Jaron, Daniel T. Berry, Julie Payette, and Valery Ivanek Takarev. It's from NASA. Okay, so the 45th spacewalk in Space Shuttle history and the 4th of the ISS era lasted 7 hours and 55 minutes, making it the longest ever conducted. And it talks about what they transferred. If you go to the NASA government site, you can read up about it. And I found scientists and payload developers can get more information about cell culturing from the payload's office. That's their phone number. And I already read that. To Earth, so I pulled that out. The cell culturing cell cult tests cells for their responses to gravity. For most cells, this means testing the role of extracellular transport phenomena, movement of chemicals across the cell wall, and the presence and absence of gravity. The cells may be cultured in suspension or attached to microcarrier beads, which provide a surface for more normal cell growth. Control over vessel rotation, which is initially required to maintain the cells in a suspended state before launch, is necessary to study the relative importance of mixing, diffusion, centrifugal force, shear, and sedimentation on cell growth. So I grabbed some pictures because I didn't know what it was. These are the cassettes that they hold the specimens in. This is a bone mac experiment made in the cell cults, made in the cassette. And it says each cell cult cassette contains a single 50 milliliter bioreactor that accommodates cells of all types in aquatic organisms. The bioreactor has options for low speed rotation, perfusion, oxygenation, and scheduled sampling. Any kind of aqueous suspension can be placed in the reactor vessel. Okay, so that tells you a little bit more about that. I'm not going to go into it. You can read it on the website there. This image shows the group activation packs, the GAPs, that house the specimens for the NLP cells 1 for their on-orbit operations. And this is the BioServe Space Technologies, University of Colorado. Okay. The facility operations, the cell coil is installed in the vehicle, usually with the reactor vessel rotating to maintain cells in suspension. Upon achieving orbit or transferring to the International Space Station, each rotating vessel is set up to off or to a speed specified by the investigator. And it operates on a programmed timeline during the planned on-orbit experiment. And then they bring the samples back to the ground to see what's happened with them. So the next thing I was Google NIHMED because I didn't know what it stood for. It stands for the National Institute of Health, and it talks about the biomedical research that they set up with the space station, and that's what all this was, the cells and stuff that they found up there. They gave them a grant. And the National Laboratory at the ISS provides a virtually gravity-free or microgravity environment where the cellular and molecular mechanisms that underlie human diseases can be explored. They're also doing testing on how bones in the immune system can get weak. And let's see what else we got here. It's their way of getting scientific information. And then GENIA corpus was the next thing I found. Provision of reference materials for the development of biotext mining systems. GENIA corpus is a collection of biomedical literature. It has been compiled and annotated with the scope of the GENIA project. I had a hard time finding stuff about the GENIA project. I guess it's so new. It's from 2012, something they just started doing, and there's not very much about it. The goal of the project is to develop text mining TM systems for the domain of molecular biology. The GENIA corpus has been developed to provide a reference material for the development of bio-TM systems. The corpus currently contains 1,999 Medline abstracts which were collected using the 3 mesh terms, human, blood cells, and transcription factors. The corpus has been annotated with various lengths of linguistic and semantic information. The next word that came up was ontology. The aim of the GENIA relation ontology is to provide a set of relations which define a detailed and broadly applicable set of relation types based on accepted domain standards, concepts for use in corpus annotation and domain information extraction approaches. I don't know what that means. So here's the axioms, relations, some sort of properties of the relations of the GENIA relation ontology. That's what it looks like. Publications. Again, it's www.tsujii.is.u-Tokyo. So it's from Tokyo. GENIA's got something to do with Tokyo. Okay, so line 18, a3a. These are the tags you'll find and what it's going to talk about. Okay, and that's for me to put on there. Yeah, just all those little words there. And then we go back over to this side here. So February 2nd, 2012, 10:31 p.m. Eastern Standard Time, my thoughts for line 18A, 3A. I learned about how many types of missions that NASA does. I couldn't believe how many there was. There's like hundreds of them I've come across so far. It's testing cells up in outer space to see what they can do to prevent bone density loss and create a better immune system. It looks like it weakens the immune system when they are up in outer space. I know I came across some documents of astronauts that have problems with their knees, muscles, and walking because of the loss of muscle mass. Now I know why I'm taking a test pill for blood pressure and muscle buildup. It's probably the same thing they use on the astronauts to see if it can help them. On Earth we have the pull of gravity and walking with that pressure working against us. What can we add to the body to give it pressure so it feels like it has to work hard to keep moving? I noticed that in the Mars 500 there is a gym to work out in. Not sure what that's for or if they've actually developed a place for them to work out. Why don't they create a large room that has a gravitational field in it so they can work out in it and keep their muscles lean and in good shape? I think we'd have to find something that can make gravity for a small living space. I'll Google it to see if anything comes up. This is what came up. Artificial gravity is the varying increase or decrease of apparent gravity g-force via artificial means, particularly in space but also on the Earth. It can be practically achieved by the use of some different forces, particularly the centrifugal force, fictitious force, and for short periods linear acceleration. The creation of artificial gravity is considered desirable for long-term space travel or habitation, for ease of mobility, for in-space fluid management, and to avoid the adverse long-term health effects of weightlessness. And this is a proposed Nautilus X International Space Station centrifugal demo. This is something that some tests that they did on this thing here. And the test done was without G-force, space adaptation syndrome occurs in some humans and animals. Many adaptations occur over a few days, but over a long period of time, bone density decreases, and some of this decreases may be permanent. That's kind of scary. The minimum G-force required to avoid bone loss is not known. Nearly all current experience is with G-forces of 1 G on the surface of the Earth or 0 G in orbit. There has been insufficient time spent on the Moon to determine whether lunar gravity is sufficient. A limited amount of experimentation has been done by Dr. Alfred Smith of the University of California with chickens, since they are bipeds, and mice experiencing high G-force over long periods in large centrifuges on the Earth. Rats have been exposed to continuous artificial gravity of 1 G during Russian biosatellite missions lasting 2 weeks. The muscle and the bone loss in these animals was found to be less than rats in 0 G. Astronauts were exposed to artificial gravity levels ranging from 0.2 to 1 G for a few minutes during several spaceflight missions using linear sleds or rotating chairs. They did not perceive any changes in their spatial orientation when the G level was lower than 0.5 grams at the inner ear level, where the sensory receptors for gravity perceptions are located. I didn't know that. Isn't that cool? We have little receptors in our ears for gravity perception. Did you know that? I didn't know that. Now we know why aliens have those little things sticking out of their heads. That's for their gravity perceptors. That's my own little joke. Anyways, since this is a big topic that needs addressing, I've moved the search data for artificial gravity to line 18A 3D. Actually, I've moved all the lines around, so I don't even know if that's true anymore, but it goes from line 18A 3D, and it's all up to F and H right now. I still have a lot more research to do. So, if you want to take a look at the next video, line 18A 3D, NASA create artificial gravity UFO engine design. I think I've changed that title too. This is from 1969. This is what NASA was thinking about doing for artificial gravity space station. This is an artist concept that they drew. I thought that was kind of cool. So that's the end of that video, and we're going to go on to the next one.